脑人言，
我是赵振东，我现在在上海交通大学医学院松江研究院工作。今天呢，我们非常荣幸能够邀请到这个哈佛的 Medical School 的 Fresh PhD 齐立军博士。然后，齐立军博，我认识立军博士，因为我在那个哈佛 Medical School 做 postdoc 的时候就认识立军博士。齐立军呢，反正他给人的这个印象啊就是你跟他接触过之后。你就能非常清楚，这是一位他具有所有很多优秀科学家的一个重要的潜质，聪明、勤奋，而且谦虚，就是为人也是非常的 nice。然后秦立军博士呢，他在13年到18年在北京大学医学部本科毕业，随后就中间没有 gap 去这个 Harvard Medical School 读 PhD，后来去了 David Ginty 那 David Ginty 呢也是在这个 sensory，尤其是 touch sensory 这个领域里面非常应该是极其 top 级别的这个 scientist，而且现在呢也是 Harvard Medical School Neurobiology Department 的 chair 的系主任。然后今天呢，秦立军博士来给我们分享他的一个最近发在这个 Cell 上的一个文章，就是他们制造了很多根据 single cell sequencing 的结果，做了很多 DRG neuron 的这个不同的细胞类型的这个工具小鼠，然后来非常系统，而且非常脚踏实地的来研究这个 morphology 和这个 function 和这个 gene 之间的这个 genetic marker 之间的这个相互关系。那么我就不多说了，我把这个时间交给秦立军博士。李军，你要开麦吧？我看你麦没开。
非常感
嗯
谢，非常感谢邀请，然后非常感谢这个介绍，我觉得就是完全是比不上这个郑东师兄的介绍，呃呃，就是因为这一次很多就是 talk 里面的很多词我都不太会翻译，所以说我可能这一次 talk 还是主要用英文讲，但是之后的Q&A 环节我可以多用中文回答一下。
嗯
Okay, so in this talk, I want to talk about a DRG genetic toolkit that reveals the morphological and functional diversity of somatosensory neuron subtypes. Somatosensation is important for us to sense texture, for social interaction, for sensing noxious forces, and for sensing different temperatures. The first order neuron that sense somatosensory stimuli are those root ganglion neurons. The DRG neuron have two branches. The central branch innervates the spinal cord and brainstem. The peripheral branch innervates different types of skin and internal organs. A classical study classified the different DRG neuron subtypes based on the physiological properties. The pioneers in this field could record from the sensory fibers that innervate the skin regions. Some of the neurons they recorded have low threshold when they respond to mechanical stimuli, and this type of neuron are called low threshold mechanoreceptors. Here, the mechanoreceptors refers to a type of neuron, not a type of molecule. And these type of neurons are very sensitive to light touch, such as air puff, brush, or vibration applied on the skin. They are also high threshold mechanoreceptors. They are less sensitive to mechanical stimuli, and they have higher threshold. They are also neurons sensitive to different temperatures like cold and heat. They are called thermosensory neurons, and they are also neurons sensitive to both mechanical, thermal, and chemical stimuli, and they are called polymodal neurons. These are the physiological defined cell types that have been known since 1960s. To understand the other properties and functions of these cell types, we need to link their molecular profiles with their physiological properties. One important strategy is to use genetic labeling approach. In this example, the Cre or Cre-ER molecule is controlled by the expression of the marker that can label certain DRG neuron subtypes, and it can be crossed to the Cre dependent reporter line, or we can use Cre dependent virus to express fluorescence protein or opsin in these certain type of DRG neurons, so we can study their properties and functions. In this example, our lab generated TrkB Cre-ER line because TrkB is specifically expressed in certain type of DRG neurons. And when we cross the TD tomato reporter line with the TrkB Cre-ER line, we can express TD tomato protein in the TrkB positive neurons. And in this example, it beautifully recapitulated the TrkB positive neurons in the DRG. We can also check their labeling in the skin. And this is labeling in the hairy skin. And each circle here refers to one hair follicle. So one neuron can innervate dozens of hair follicles, and they form these interesting lanceolate endings around hair follicles. We can also record from the TD tomato positive cells. And these type of neurons are very sensitive to hair deflection. And we can also perform activation and ablation studies to check what are the behavioral phenotype when we activate or silence these type of neurons. Other than our lab, Dr. Xin Zhongdong, Qiufu Ma, Zhou Fengchen, and other labs also did great work in the genetic labeling of DRG neurons. Meanwhile, there is very high diversity of transcriptional profiles of DRG neurons revealed by the single-cell RNA-seq of DRG sensory neurons. In our recent work, Dr. Nikhil Sharma, a former postdoc in our lab, did a single-cell RNA-seq of DRG neurons and found there are more than 15 clusters. Some of the clusters are known and can be labeled by the existing mouse lines. One surprising finding is the diversity of CGRP subtypes. CGRP is a common marker for labeling nociceptors. Previously, people take a DRG neuron more or less as one cell type, but in the transcriptional studies, we found CGRP neurons have more than 7 subtypes, and most of the subtypes were unknown before. To label the new CGRP subtypes, we generated new driver lines using the molecules that are specifically expressed in these CGRP subtypes. And there are also DRG subtypes that were known before, but without very good genetic tools. We also generated driver lines to label these subtypes. So to validate the specificity and efficiency of the newly generated lines, we crossed the driver lines with Cre or Philippe dependent reporter, or we injected a Cre dependent virus into the animals. And we performed RNA scope to validate the specificity and efficiency. The x-axis here are the specificity, which means the neurons that express, like the percentage of neurons that express the reporter, whether they also express the molecule. And the y-axis is the efficiency of the mouse lines. And these lines pass the quality control. So combined with the existing driver lines, we assembled a genetic toolkit for DRG neurons, allowing us to systematically investigate the properties and functions of DRG neuron subtypes. In this study, we are interested in answering the following questions. What are the morphological features of distinct DRG neuron subtypes? And how is the mechanical and thermal information encoded by the distinct DRG subtypes? And this project is a close collaboration with Dr. Nikhil Sharma, a former postdoc in our lab, and a current assistant professor in Columbia University. We want to test the hypothesis that transcriptionally defined DRG subtypes have distinct morphological and physiological properties and functions. In this study, we focused on the hairy skin that covers the most part of our body and of the mice body. There are different sensory structures in the hairy skin. Some are associated with Merkel cells, and some form the lanceolate endings around hair follicles. Some form the circumferential endings, like a circle around hair follicles. And the previous study from our lab and other labs already identified the DRG neurons that form these type of endings. In our new study, we found more than 7 subtypes, or most of the DRG subtypes, form free nerve endings in the hairy skin. To better understand the morphological features of the DRG subtypes, we performed sparse labeling to characterize the single neuron morphology. So we perform whole-mount alkaline phosphatase staining, which can stain the nerve fiber in purple in the transparent or white background, very easy to visualize under the bright field microscope. To express AP sparsely in different DRG neuron subtypes, we cross the Cray-ER lines, which can be induced by tamoxifen to brain-straight AP allele. So the AP protein is expressed in a Cray-dependent way and under the brain-straight A promoter. And brain-straight A is specifically expressed in the sensory neurons in the skin. By using a titration of tamoxifen, we can label very small subset of different DRG neuron subtypes and achieve the single neuron morphological analysis. For the Cray lines, we injected a titration of AAV that is Cray-dependent, that express alkaline phosphatase. So in this example, the sparse labeling of tract B population, we can see one neuron can innervate dozens of hair follicles. It can be confirmed by the fluorescence labeling. But in this case, we only do the alkaline phosphatase staining, and we actually reconstruct the morphology formed by single neuron. So this type of neuron are associated with Merkel cells, only innervate one hair follicle. I don't have the zoomed in image, but it has been well established. And these 3 subtypes form lanceolate endings, and each neuron can innervate dozens of hair follicles. And it's also been known. Previous study also found there is this type of neuron, and each neuron can innervate up to 100 hair follicles and forming the circumferential endings around hair follicles. Our new population, labeled by the BMP-R1B gene, we found one neuron can innervate even more hair follicles and forming circumferential endings. In this quantification of the number of hair follicles innervated by the different DRG subtypes, we can see the BMP-R1B neuron innervates the most number of hair follicles, up to 300, being the largest among the hair follicle-associated endings. We also found there is great morphological diversity of free nerve endings. Previously, free nerve ending is taken as one type of sensory structure, but we found there is actually great diversity in their branching pattern and in their innervation areas. SMR2 neuron has very sparse branches, and it can cover a very large area, can innervate up to 33 square millimeters. On the other end, MRGD positive neurons have this very dense branching pattern, forming this bushy-like ending, and their innervation area is much smaller. If we quantify the branching density, we can see there is great difference of different DRG subtypes. SMR2 neuron only have 1 or 2 branching points per square millimeter, where MRGD neuron have more than 1,000 branching points per square millimeters. So there is great morphological diversity in their branching pattern. We also compared the innervation areas. In the left of the dashed line are the hair follicle associated endings. In the right of the dashed line are the free nerve endings. We can see certain neurons are really small. They only innervate 1 or 2 hair follicles, and SMR2 neuron covers a really large area. So there is great morphological diversity of the DRG neuron that are genetically and morphologically defined. There is one population that we didn't see any innervation in the hairy skin. At first, it is ADR2A CRE-ER line. At first, we thought this CRE-ER line is not efficient. Then when we checked the labeling of the DRG, we found there is actually very interesting pattern. There is no labeling in the lumbar or cervical level, which are the limb level, but there are more neurons labeled in the lower thoracic and in the sacral level. And in these levels, there are usually neurons that innervate the internal organs. So we checked the labeling of the internal organ. For example, bladder, we found there is very dense innervation of bladder. So although other DRG subtypes might also innervate internal organs, the ADR2A population is the only population that exclusively innervate internal organs. What I showed are the skin terminals of the DRG subtypes. Next, we want to look at the spinal cord terminals of the genetically labeled cutaneous DRG subtypes. We performed cryosection and immunostaining for the spinal cord dorsal horn. For trigeminal neuron, terminus very superficially in the spinal cord. Here, compared to CGRP, which labels the lamina 1 of the spinal cord, and AB4, which labels the lamina 2 of the spinal cord, the termination of trigeminal neuron is even on the top of CGRP, so it's on the very surface of the spinal cord. And these three populations are all CGRP positive. They all overlap with the CGRP band. Interestingly, the BMPR1B and SMR2 population, they also send branches down to the deep dorsal horn. So very different from SSTR2. And by the way, all the names in this presentation are not important for you to understand the conclusion of the paper. And we found these 4 populations have spinal cord terminals located in the AB4 layer. And the CLTMR have spinal cord terminals below the AB4 layer. And these 2 A-fiber LTMR subtype have terminals even deeper than the CLTMR's terminals. If we compare all the spinal cord termination patterns with CGRP and AB4, we can see there is distinct spinal cord innervation patterns by the different DRG subtypes. Next, we want to study the physiological properties of the distinct DRG neuron subtypes. And we performed in vivo DRG calcium imaging. Because the DRG is located next to the spinal cord, we had to anesthetize the animal and remove the bones and muscles on the top of the DRG so we can directly image the surface of the DRG. Here we choose L4 DRG, and we apply stimulation on the thigh of the mouse where the L4 DRG innervate. To express GCAM, we still use the CRE or flip dependent reporter allele or inject the AV flip GCAM to express GCAM in the distinct DRG populations. In this example, we crossed TH-CRE-ER with AI148, which is a population that which is a reporter line that express GCAM in a CRE-dependent way. So in this video, I am gently stroking the hairy skin of the mouse. This population is known to be sensitive to the gentle stroke of hairy skin, and I'm stroking different parts of hairy skin. So we can see different group of neurons are activated. Using similar approach, we can identify the receptor field of the DRG neurons and apply precise step indentation to the center of the receptor field from 1 millinewton to 75 millinewton. 1 millinewton is very light mechanical stimuli, and 75 millinewton is pretty intense for the mouse. To the same area, we also applied temperature stimulation from 5 Celsius to 55 Celsius using a custom-made Portier device. Let us first look at the responses to step indentations. In the first example, I want to show the response of A delta LTMR. The straight traces here are the calcium signals responding to 1 to 75 millinewton indentation. And the neuron responded to the low forces. So it is a typical low threshold mechanoreceptor. If we summarize the number of neurons with certain threshold, most of the neurons respond at 1 to 5 millinewton range. So it is a low threshold mechanoreceptor. In this example, the MRGD neuron also responded to relatively low forces, but it showed the gradient response. It has a weak activation in the low forces or medium forces and much higher activation in the high forces. If we summarize their threshold, it is in the intermediate range. And this type of neuron only responded to SMR2 positive neuron only responded to very high forces. And their threshold are only in the high threshold range. And this type of neuron labeled by SSTR2 CRE-ER don't respond to any mechanical stimulation, but they respond to other stimuli that I will show later. If we summarize the percentage of neuron with certain threshold and plot in a heat map, we can get a heat map like this. The intensity of color block refers to the percentage of neurons with certain threshold. We can see the different DRG neuron subtype have distinct mechanical threshold, forming the continuous distribution of threshold rather than a bimodal distribution of low threshold and high threshold. Some of the LTMRs are really sensitive, like these two, while some are less sensitive, but all LTMRs subtypes show the saturated response at high forces, which is not shown here. There are subtypes with threshold in the medium range, and subtypes only responding to the high forces, suggesting the population coding of mechanical stimuli by DRG neuron subtypes. We also applied thermal stimulation to the same area of skin. Shown here are the calcium responses to the cooling and heating of the skin. Because that is a lot of data, I will show a few interesting things that we got from analyzing the calcium responses. If we quantify the thermal threshold of distinct DRG neuron subtypes, we found the thermal space is also tiled by the thermal threshold of distinct DRG subtypes. Some DRG neurons don't show any response to thermal stimuli, and some neurons only respond to heat up to 50 or 55 Celsius, and some neurons responded to warmth stimuli from 30 to 40 Celsius. And trypamide neuron is very sensitive to any type of cooling. Just a few degrees of cooling can robustly activate trypamide neurons. And they are also neurons sensitive to even cooler temperatures. So the temperature stimuli is also tiled by the thermal threshold of DRG neuron subtypes. On the top of encoding force and temperature, we also found DRG neuron encode relatively change of forces. If we increase the duration of step indentation, we could start to see the adaptation properties to step indentations. For the SA1 LTMR, they showed the sustained response to step indentation, while for this A delta LTMR, they showed transient responses at the onset and offset of the step indentation, so they are rapid adapting. And all the neurons with a high threshold showed the low show the slow adapting responses. For the temperatures, we found similar things. For CLTMR, they responded transiently at the temperature decrease, shown here. And a subset of TRPM8 neuron showed the sustained response to temperature decrease or cooling. And another subset of TRPM8 neuron showed inhibition by worms, shown if we can see that the worms actually make the calcium signal go down, suggesting there is a spontaneous activity in the baseline, and TRPM8 neuron might be able to encode both cooling and warmth temperatures. And some neurons, some subtypes also encode the transient relatively increase of temperature, like MRGB4 neuron. They respond to heat, and some of them also responded when the temperature goes from 5 Celsius to baseline or from 10 Celsius to baseline, shown here. So DRG neuron subtypes encode both absolute force and relative changes of force and temperatures. Because we deliver the mechanical and thermal stimuli to the same piece of skin, we can see whether a neuron prefers a mechanical or thermal stimuli. For this subtype, they only responded to mechanical stimuli. So all the neurons here are in the mechanical axis, and each point refers to one neuron that we imaged. And for these three subtypes, they are also mostly mechanical sensitive. For CLTMR, some of them are cold sensitive, but the response to cooling is much smaller than mechanical stimuli, so they are still located near the mechanical axis. And these five subtypes are mostly polymodal neurons. As we can see, the points of these neurons are located mostly between mechanical and heat axis, so they responded to both mechanical and thermal stimuli. And this type of neuron only responded to cooling stimuli, and this type of neuron only responded to heat. So in summary, from the transcriptional heterogeneity, we made new genetic tools, and combined with the previous ones, we built a genetic toolkit that allowed us to reveal the morphological diversity using sparse labeling, which also reveals the distinct mechanical threshold and thermal threshold of different DRG subtypes. The genetic tools links the morphological profiles to, links the transcriptional profiles to morphological and physiological properties. It also enabled the functional manipulation of the new DRG subtypes to study their functions in touch and pain, which our lab are currently working on. We are also interested in the innervation of other skin regions or internal organs by the new DRG subtypes. Another postdoc in our lab, Rachel Wolfson, used the different mouse lines to identify neurons that innervate the distal colon of the mouse. In another project of mine, I found two of the DRG populations innervate the sex organs, and they mediate the mechanosensation of mating behavior. We are also interested in studying the remaining subtypes, because in our current toolkit, we didn't cover all the DRG subtypes, and there are still neurons that we haven't been able to get access to. And of course, DRG subtype is an ongoing investigation. There is always new subtypes that can be found. So in the end, I want to thanks to my mentor, David Gindy, who is very helpful and very supportive, and my great collaborator, Nikhil Sharma, and his new lab in Columbia University, and other people in our lab in the community. And thanks for listening. Welcome questions.